Okay, so we are going to start with looking at chapter 9, which is muscles on here. So muscles are there's three different types. If you remember from earlier in the semester, you have the skeletal muscle, cardiac muscle, and the smooth muscle. Most of this chapter really kind of refers to or is going to talk about more the skeletal muscle when we get into the physiology of it, just because that's kind of the the action that occurs is through the skeletal muscle. And then the others pretty much do the same thing. There's some minor changes on how the physiology of the other types of muscle work. And I'll bring that up specifically at the end. But we're going to talk about the three types of muscle, and then we'll talk about how muscle contraction actually occurs, so the physiology of muscle contraction, and then with skeletal muscle, and then look at the others in retrospect as to what the differences are. All right, so let's start with sort of a review of the muscle tissues. You did muscle tissues at the beginning of the year, so this is nothing new to you. The three types are skeletal, cardiac, and smooth. The first one we're going to talk about here is skeletal muscle. Skeletal muscle is found attached to the skeleton. That's where it gets its name. So it has that origin and insertion and portions that allow for the muscle to pull against bone and allow for movement to occur. So we do see it as attached to bones. When we look at skeletal muscle, we have terms, or any muscle, I should say, we have terms that you guys need to start getting to know. First one is voluntary. So skeletal muscle is the only muscle type that we consider voluntary. And what that really means is you have this conscious control over it. You determine, I want to stand up. And when you determine I want to stand up, then your brain sends nerve impulses down to sets of muscles and said, it's time to get to work. It's time to start contracting and doing these different actions to allow for the not only the muscle to contract to pull on the bone so you can stand, but other muscles need to relax more. You need to have this organized movement so that the joints stay in place and everything moves together in a sort of a smooth fashion. But it is this voluntary action of saying, I want to do this now. So that control over that, or I don't want to move my arm at this moment. So you don't. The other terms that you need to get to know, there's this term called somatic and autonomic. Somatic, always think of it, these two as going together. Somatic really means like body. So when we hear like somatic cells and stuff, we're talking about body cells. But anything that's not viscera, so anything that's not part of the organs really is somatic. But somatic tends to be associated with voluntary, okay? Remember that when we looked also at the tissues, you have muscle tissue are these long thread-like cells. And when you look at them under the microscope, they have these little bitty stripes that go across like this. Those are called striations. So muscle cell is striated, and that was one of the key factors that I told you to look for to tell the difference between muscle cells, or some muscle cells, these muscle cells, and connective tissue that looked very similar to that. Also, muscle cells tend to have very many of these, dark purple in here, a lot of nuclei per cell. So these long strips are one single cell, and you can find many nuclei pushed off to the side for that. So that's referred to as multinucleated. Cardiac is the second type of muscle tissue that we have, and that's found strictly in the heart. So that's just a heart thing. It's considered involuntary, and it's considered involuntary because you don't have that conscious control. I can't say stop and start. It's vital to your existence. So from the time very early on in embryonic development, when cells start to differentiate and you start getting tissues and you start forming these organs and these different tissues that will form organs, the heart cells begin to contract. And that continues all the way till the last breath you take. So the last part when the heart stops contracting, because it's such a vital thing. You have to get blood around to the cells in order to give them nutrients and get rid of waste products. So this is considered an involuntary movement, or involuntary control over this organ. You don't have that choice in the matter. So again, words that you need to associate together. Involuntary is autonomic. And when you hear the term autonomic, what you need to think of is automatic, okay? Autonomic is automatic. In other words, you don't have that control over it. The only control you may have over this muscle is relaxation techniques. So you can say, my heartbeat's a little fast, or I'm a little hyped up right now, so breathe in. So you take these deep breaths, or you have these meditation techniques, or whatever it is that will allow for the heartbeat to slow down and give you some sort of relaxation. But you can't stop it, and you can't restart it on your own. So that's why it's autonomic. When I told you at the beginning of the semester how to look at cells and recognize these cells, again, cardiac's like skeletal in the fact that it's these long threads, but unlike skeletal, cardiac tends to branch. So you have this branching going on in here. We do have the striations, so you can see these little stripes going here, little striations, so it is striated. But importantly, cardiac has these intercalated discs, and that is solely specific to cardiac muscle. So remember that. All right, smooth muscle. Smooth muscle is called smooth muscle because there are no striations. So this one doesn't have striations. There's none there. The cells tend to be long and thin like the other cells, but unlike the other muscle cells, they're more tapered at the end. So you have these long cells that get wide in the center, and then they taper out at the end. And they're single nucleated, one single nucleus there. And I drew them a little fatter than you see here, but you can kind of see starts and stops. So here's the end of that one, and it comes down in that way. Again, this is an involuntary type of muscle, and it is autonomic. So you find it in things, it says viscera here, but like your intestinal organs, so the gastrointestinal tract, blood vessels and skin, and we'll talk more about that later, but like esophagus, that's the first point of contact with when your food or your water or something comes into contact with your gastrointestinal tract, where you come in contact with smooth muscle. So you take in food, you chew the food, all of that is still voluntary. That's skeletal muscle that's in control of that. Even the swallowing portion, I swallow that food, that is a voluntary action. But as soon as that action has occurred and it moves past your pharynx into that esophagus, that's when it's going to start coming in contact with the smooth muscle. And really what's happening is you have this, so if this is your gastrointestinal tract going down, and this is your esophagus going down, soon as that food comes in contact with that muscle, it kind of stretches that muscle. So it causes this bulge here, and it stretches that muscle. That stretching of that muscle causes something called peristalsis to occur. So peristalsis is this rhythmic contracting and relaxing of the muscle that really just stretches and then relaxes and then stretches a little bit further down and then relaxes and then stretches, and it's pushing that object, food, water, whatever, down towards the next organ. Okay, so it is autonomic. All right, so looking at muscle, each muscle is an organ. And remember, by definition of an organ is made up of several different types of tissue. So we do have muscle tissue. That's very important. Otherwise, it wouldn't be muscle. But you have nerves that run to that and allow for control over those muscles and directions to those muscles to give them directives to do something. You have to have a very rich blood supply because you got to bring in oxygen, you got to bring in glucose, you got to take away waste products. So you have this very rich blood supply that is going to feed that. And that's really what gives it that pinkish color too, by the way. So it's the rich blood supply and the types of tissues. And then we have connective tissues. We have all kinds of tissues that are binding these together. And you can see over here the white in here. So binding from muscle to bone, like in the tendons, binding, you'll have these, you can, like on his stomach right here, you can see the white right in here. So binding muscle to muscle, that kind of stuff. Those are all connective tissues. One of those is called fascia. So fascia are these long sheets that usually surround the bone and lead into, it's always a continuous thing. So it's like muscle, fascia to binding to something else. Tendons, I mentioned, that's binding muscle to bone. And then these things called aponeuroses. Aponeuroses are found in large sheets also. So you have these sheets here, but there's specifically, you have one epicranial on top of the head, and you have the one, the abdominal across the stomach in there. And you can see little pockets of muscle, a little six pack going on in there. And the aponeuroses is kind of that sheet that binds all that together. Here's a tendon. in this picture. So here's the muscle, here's the bone, tendon here. But notice I told you it was continuous. Here's muscle, and you can see this sheet that's starting to turn white. That's fascia that then becomes the tendon. So it's this connective covering over the muscle that allows for the tendon to do its job also. And then as we look, you can see here's my organ, the muscle, right? So the muscle is the organ. And then I'm going to get smaller and smaller and smaller. It's kind of like if you think about a rope that holds something, maybe like a ship's rope or a boat's rope. I guess a ship would have metal, but anyway. It's not just one big solid piece of material. It's this bound, bundled section. So I can take the rope and I can untwist it. And when I untwist it, I get a smaller section. And then I can take out a smaller section and a smaller section. That's what your muscles do. And basically, when you get down to it, one small thread may not be that strong. You know, think about even threads on a spool versus a blanket that's made up of a whole bunch of threads. But if you put them all together and you start binding them and twisting and then getting bigger, binding and twisting, it becomes this very strong organ in the end made up of all these smaller pieces, okay? So the first level from muscle down is what we call a fascicle. So this fascicle, let me do some erasing here. So here's the muscle, that's the organ. Each little circle that you see in here, that's a fascicle. You can pull that fascicle out. So here's one fascicle here. And you notice that it's also made up of these smaller bundles itself, okay? So then we have getting smaller and smaller, and you're going to end up with the muscle fibers, which are actually the cell. So when you hear fiber, think cell. And then it gets smaller, and you can see right here, here's a myofibril. So that's a section of that. We'll talk about the blue and the pink and everything in here in a minute. And then you have these smaller portions called myofilaments. And the myofilaments are two very specific, there's more, but two very, sorry, lost my train of thought. Myosin is one of those filaments, and it's protein. And then actin is the other main filament that make up the actual contracting unit. They contract together. So they work together to form a contraction in your muscle. All right. So again, terms. Everything has sections and everything has little things. So around the outside, here's the whole muscle, right? So that's the organ right there. Around the outside, we have what's called the epimysium. These are just coverings. So these are thin coverings around the outside. So this white that goes around the entire thing right here, okay? The entire section. You have the next one, so each little section here, if I pull that out, so inside, so around the outside was the epimysium. Around the inside here, I have the perimysium, and then I pull a smaller section out, and I have another little bundle that holds it together that is in between these smaller sections, and that's called the endomysium. So epi on the outside, peri, and then endomysium. Remember, then you have the muscle, that's the whole organ. We have these first level of bundles that we pull out called fascicles. Muscle fibers themselves are the muscle cells. And then getting smaller, we have those myofibrils, all right? And then the smallest of them, the myosin, the myofilaments, which is actin and myosin. There's others, like I said, there's titin and there's some other things we're going to talk about. But the actin and myosin are really the two key ingredients that allow for this muscle movement to pull in, contract in. They interact. So if my fingers right here are the filaments, they interact with each other to pull in, causing a contraction, and then pull apart, and that's relaxation. So to allow that muscle to contract, they have to actually interact and pull with themselves. All right, muscle fiber. Remember, this is a cell. So we're talking about a special cell called a muscle fiber. Every cell around the outside has a cell membrane. The cell membrane for a muscle cell is called a sarcolemma. So that has a special name. So sarcolemma is the cell membrane. Notice these terms. Sarco. That's one of those things. Sarco means muscle. Okay. I did what y'all do all the time. Just misspelled it. Sarco, S-A-R-C-O, means muscle. And another term you're going to see is myo. Myo also refers to muscle. But sarcolemma, the lemma is the membrane. Sarcolemma means cell membrane or skeletal muscle cell membrane. Sarcoplasm, cytoplasm. That's the cytoplasm of a muscle cell. Okay, you got the plasm portion, and then it has sarco that refers to muscle. And then sarcoplasmic reticulum. That should sound pretty familiar to you because that sounds a lot like the ER, endoplasmic reticulum. So it has its specialized ER that we call a sarcoplasmic reticulum because it has a specialized job. It does something specific for that cell that other ERs would do differently for other cells. Key structures that are defined for muscle cells themselves or muscle fibers, something called a T-tubule. It stands for transverse, but you can refer to it as a T-tubule. Triad means three, so a triad is three. It's one T-tubule, one of these, and two of the little SRs. So two, or the cisterna of the SRs. The SR is a big, huge network, but it's these little cisterna. Down here in this small picture, I know it's kind of small, but you can see the green on the inside. This three that go across here, that's the triad. And then you see the network in between, that's the SR. I've got a bigger picture on the next page. So right here, one, two cisterna of the SR right here, okay? And then in between, I have one, two T-tubules. So 1, 2, 3, that's your little triad right there, okay? And then in between, all this little, looks like a little network of tubes, that's all part of that SR. So these sort of darker yellow right here, tubes, these are called cisterns or cisternae. And then they can connect with the rest of the SR that's in between. And basically what the SR is going to do is it stores calcium ions. So the calcium ions are really important for muscle contraction. They control whether that muscle contracts or not. So they're kind of indicators to say it's time to start getting to work. And the calcium ions are stored in the SR, and then they're released from that SR when it's time to contract a muscle. Notice also, see how the nucleus is pushed off to the side? Really what we want is most of the action for the muscle contraction to occur in these long fibers, so the nucleus is not centralized like you would see kind of in other cells. Also notice, bunch of mitochondria. What does mitochondria do? This is your powerhouse. So this is ATP, and if I'm going to have muscle contraction, I've got to have a lot of energy available to those cells at all times or at any time when required. So we have a huge amount, all these little yellow dots in here are mitochondria. So there's a bunch of mitochondria that's present. Okay, when you see these things over here, I've got these little practice quizzes and stuff. Get off of that. Oop, go back. Those are links that I'm not going to go to during this actual presentation, but when you go to my PowerPoint that I gave you on D2L, you can go to those and practice. And basically, they're little tutorials or they're little quizzes or there's something that can kind of explain a little bit better. A lot of them are little animations that show it instead of me drawing it because my drawings are so good. It shows the actual action that's occurring for these things. All right, so let's start talking about, again, key players, and then we'll get into how that muscle contraction actually occurs. The myofibril is that smallest little section of the muscle, and the myofibril contains myofilaments. The two main types of myofilaments, once again, are actin and myosin and their interaction. Now, within that, so we're going to have these actin, and we're going to have this myosin, and they're going to, I'm just going to draw like two here. So they interact like my interlocking fingers. So I have this myosin right here, and I have this actin, and we're going to have them kind of interlocking like this. They have units that control the contraction. And you can see in that striped look that you can see when you look at the muscle tissue under the microscope. These little stripes, the striations, are these portions of this contracting unit. So one contracting unit, that we're going to spend a lot of time talking about, of a muscle cell, so this is a contracting unit, is called a sarcomere. So sarcomere, again, sarco refers to muscle, but that's a contracting unit. So one specific unit that allows for contraction. You've got millions of these guys working together just for one contraction. All right, here's our two different types of fibers. So, and I told you there's other types in here too. So you got to kind of realize that there's two main, actin and myosin, but there's going to be some other guys, other players that you need to get to know also. So myosin, they've got it purple here, but it's different colors for different diagrams. But purple here, the myosin is what we call the thick filament. Notice in relation to the yellow, it's a lot thicker. The actin is then what we refer to as the thin filament. And it really is a bunch of little protein spheres, these little balls right here, that are linked together in chains. And you have these two chains of these spheres that interact and form that actin. Surrounding the actin, you have these long thread-like proteins. So, and I've got two of those also, right? That is called tropomyosin, okay? Myosin itself is the big, thick, and it's long threads that are all bound together. And then in addition to that, I have what they have in this diagram, these little green proteins spread out. That's called troponin. So the two interactions are going to occur between actin and myosin, but the troponin and the tropomyosin are really, really important because without them, you couldn't have the combined action of a contraction and then a relaxation of that. This top picture up here that you see with these little two heads coming off, that's part of the myosin. So that's what they're showing down here in these little heads. You notice they can extend out or they can relax backwards. So their action in here. What's going to happen in a contraction is those myosin heads, these two heads here, have to be able to come in contact with the actin, those little spheres. So during a contraction, you have to have the ability of a binding action between those two. In relaxation, we don't want them to bind because we don't want them to pull on each other. So that's where the troponin comes in. So as long as the troponin is on the outside of this filament and the actin is faced towards the center, see how the actins, the troponin's on the outside and the actin's on the inside? These myosin heads have no way of interacting with the actin. So there's no contraction. So in order to get a contraction occur, I'm going to have to reverse this action and pull these little spheres on the outside and pull that little thread to the inside so that the sphere's on the outside. And when that occurs, then these little myosin heads actually can attach to those spheres. And when they do, it's like an oar stroke. If you've ever paddled, you paddle, so it paddles backwards. So it attaches to the sphere, it paddles backwards, and then it releases, and the myosin head goes back into its place, and it picks up another one, and it pulls it. So basically what it's doing is it's just forcing that actin filament to move in one direction. And then when it relaxes, it stops holding on to it, so the actin moves back again to its original position. All right, so again, it's going to be the movement of those two filaments, the actin and myosin, in relationship to each other that causes a muscle contraction. Talked about the sarcomere. So when we're looking here, we're looking at sarcomeres. Sarcomere is that contracting unit, so that's something you have to keep in mind. And I'm going to start referring to something on here that's called the sliding filament theory. The sliding filament theory is the action of the sliding of that actin across that myosin that allows for a contraction to occur. Okay, so this explains, it's the model of how we actually explain how a contraction occurs. So in order to look at this, we're going to have to look at different things. So from this point right here, see that purple line, that's zigzag, to this purple line right here, that's zigzag, and then you would have another one down. That's one sarcomere. So that whole section right there is a sarcomere. What's going to happen is these two little, what we call, they look like zigzags, they're called Z lines, okay? So those are the Z lines right there. Those Z lines will move closer together. And the reason they move closer together is the actin moves inwards towards the center, towards each other. So the actin on this side and the actin on this side actually come closer together. All right, other things to look at. So this right here, here's the Z line coming down. Here's the Z line coming down here. That's one sarcomere, and then this would be the next sarcomere on this area. But so in between here, I'm going to have these different areas: I band, A band, H zone, and M line. Okay, that gets confusing. So you have two bands, I band and A band. You have an H zone, and then we have this M line. Let's start with the M line just because it's easier. The M line is the center. See these little spheres they have right here? The very center of that sarcomere right here is the M line. So that comes straight down the center. That's your M line. M for myosin, because the only thing in that area is myosin. Okay, there's no actin in that part right there. This light pink in the background, I'm sorry, that this in this diagram, this darker, I don't know, lilac color right here going across, that's myosin. The light blue that you see here, that's all actin. Okay, so in the middle, all I don't have any overlapping actin. None of this blue color right here doesn't come all the way to the center. Only the myosin in the center. So that's the M line. Then we have the I band. The I band, and you can see here on this diagram here, the I band consists of only right here to here. So it's in between right here and here. If you look, you can only see the little blue color of the actin. So there's only actin interaction going on in here. M line is myosin. It's in the center of a sarcomere. Okay. I band is this is actin. Notice I've got the end of my sarcomere right here. So the I band really consists of the actin from one sarcomere all the way through the actin of the neighboring sarcomere. And then we have the next one, which is the A band. A band is an overlapping, and you can see it right here. It's here to here, and by the colors, you can see they've got this lighter purple and this darker purple that's going on in here. So I have myosin, and I have some actin overlap. So here, see the actin right there? There's the actin coming in, and then I have these thicker myosins going across. So there's this overlap here. So it's really from the end of the myosin threads all the way through the center. So I do have a little bit of actin running into the center here of that. So there's this myosin and actin overlap going on in there, but it ends, the A band ends where the myosin ends. So it doesn't include, it does not go all the way into the rest of that actin. So it's only like part of the actin. All right. And this is going to be key, because this is going to be that interaction in there. And I don't know if I mentioned Z line. Z line was the actin, the end of the actin from one sarcomere to the end of the actin on the other sarcomere. Okay. All right. So in this picture, different colors, same thing. The middle, this is myosin. Okay. The little spheres over here, this is the actin. And remember, we have the troponin, which are their long threads, and then tropomyosin, which are those little bitty things here. Since you see the whole thin filament is that actin. Since you see the troponin, tropomyosin, I don't know why I'm saying this wrong. The tropomyosin on the outside, these little heads from the myosin cannot come in contact with the actin, so now we're at a relaxed state. Okay, those little these are called myosin heads. They're also referred to, the whole thing's called a cross bridge, because it crosses that gap and allows for this to occur. The little troponin right here is going to be, I'm going to tell you its function in a minute, but it does control the action of that tropomyosin as to whether it's hiding the actin or whether it's exposing the actin. Okay, so thick filaments, we've got the myosin. That's this across the center with these cross bridges. Thin filament is three things. It's the actin, which is the spheres, and that's kind of the functioning unit of what's going on in this contraction. And then the interaction of that troponin and the tropomyosin. So troponin, the little segments, and then tropomyosin, the long threads that cover up the actin. All right. So you say, hey, I want to move. I want to stand up. Your brain sends an impulse down to the muscle, and it's going to come in contact with the muscle fibers. So here's my nerve right here coming in, and you're getting this electrical impulse that comes in to that end. Now I have to take this electrical impulse and I'm going to have to convert it into a muscle contraction. So this is where this thing called a neuromuscular junction, or NMJ, is going to come into contact. So here's my nerve. Here's my muscle cell. Notice they don't come in contact. There's a little gap in between. This is going to allow an electrical signal that comes down from that nerve cell to be converted into a chemical signal. Those little blue dots there are little chemicals. So I have a chemical signal that is going to actually allow that information to be passed from my nerve cell to my muscle cell and then direct the activities of that muscle cell. So a neuromuscular junction is going to have a lot of things that are coming into play here. I have to have a neuron. I have to have a nerve cell. We call it a motor neuron because it's coming away from the brain and it's giving information to your body to do physical work. So motor, right? You have a motor end plate. The motor end plate is just that section there where one of those ends of that nerve comes in contact with the muscle. So this whole thing is a motor end plate. We have something called the synapse. See this gap right here that's in the blue or the gray color? That gap where the nerve does not actually come in physical contact with the cell, that's called the synapse. So that's the gap that occurs between those two portions. We're going to talk about synapse again when we talk about nerve cells and their actions. So don't forget that, don't let that slide by. That's going to come back. So it's that gap. And the cleft is the same thing, the cleft here. The cleft is the actual gap. The synapse is this interaction where we're going to have chemicals also in it. All right. And then we have synaptic vesicles. So if you're looking at, I'm going to draw this bigger, but this nerve cell right here. So you have, here's the nerve cell here. My electrical signal came down this way. Once that electrical signal gets down to the end, I'm going to have to say, hey, I can't send an electrical impulse across that gap because you can't just send it across a space, right? So now I have to convert it to chemical. So what's going to happen is you've got these gated channels, these little proteins that are normally closed. They're going to respond to this electrical signal and they're going to open up. So a little gate opens up right here. And when that happens, calcium flows in. Here's one use of calcium. Calcium flows in, and then calcium causes these little vesicles that are inside the cell that are filled with a bunch of neurotransmitters. The most common one is acetylcholine, okay? It causes that to move to the surface, down to the end down here, and then release those little neurotransmitters out into that little synaptic cleft. So then, going back over here to this drawing, so then those little blue dots that you see, the neurotransmitters, cross over and then they will bind to the muscle cell on the other side. And the muscle cell is going to have little proteins that it binds to. And when those little neurotransmitters come in here, once again, we're going to open the channel. So here's my, here's my muscle, here's my closed channel. And then when the neurotransmitter binds to it, it opens that channel and we're going to have a substance called sodium flow in. So it allows sodium to flow in. I have gone from an electrical signal up here to a chemical signal with calcium that got transmitted to another chemical signal called acetylcholine, cross that gap, and now a third chemical signal, sodium, is now going to trigger that muscle cell to do what it needs to do to contract. There's a lot of different neurotransmitters out there. The main one for muscle contraction is this one called acetylcholine that we shorten with that ACH. It's a capital A, capital C, and then a little h. It's the shorthand for that. Okay. Again, I have included this this animation in your links on D2L. But again, just to explain where we are, This is the nerve cell, electrical signal. This green is the calcium channel. As soon as that electrical signal hits the calcium channel opens. These little green dots are calcium. They flow in. They trigger these synaptic vesicles that are filled with acetylcholine to move to the surface, and they'll bind to that surface, and they release the acetylcholine, these little purple dots, out into that synapse. So this is the synaptic cleft, or the synapse right here, this space. And then the acetylcholine binds to these proteins, these little channels, opens them up, and sodium will flow in, and sodium is going to cause the first step in your muscle contraction. And that's what this little video is going to go through. So go back and look at that video and make sure that you can see that. It looks better than my drawings. All right. How motor units work, or how muscles work. You can see in this diagram, I've got like one nerve cell, or nerve axon, we call it, coming in here. But notice how it splits, and then it splits again, and it goes to a bunch of different cells. You don't have one nerve coming into each muscle fiber or each muscle cell. That would be way too many nerves in your body to do that, and it would be a huge coordination of effort that you couldn't do. It's much easier to send that impulse, or that information that says, hey, I want you to contract, down one tract, and then have that tract split into several different branches that can control a bunch of different muscle cells. That's what we call a motor unit. So one neuron, many fibers, okay, controlled by that. We can have very few. So in this case, you can see it's kind of 1, 2, 3 on here. This one has 2, but it's a diagram. In reality, you have as few as 4 fibers, and then you can have as many as thousands of muscle fibers that are controlled by one single motor neuron, which is convenient. So here's the difference. Why would I want only a few or want a whole bunch? It depends on the work that you're going to do. If you're going to do a precise movement, so think about your eyes, and take your eyes, and without moving your head, just kind of look over to the side, right? When you look over to the side, that's a very precise movement. Your eyes are focusing from one little spot to a small little spot somewhere else, okay? We don't want a whole bunch of information coming in all at once to tell these muscles, you guys all need to contract. We want just a few muscle cells to do their job to get that very distinct motion. So for precise movements, you want very few muscle cells to actually be controlled at once. For a big movement, so say I wanted to lift a desk, okay, I need to put a lot of effort into a lot of muscle cells, and I need to do, tell them all do the same thing at once. So I need to have that neuron tell a whole bunch of my muscle fibers. So a lot of force, oops, dang it, a lot of force, I can't write with this thing, sorry, I apologize, guys. A lot of force, then you're going to have to require a lot of fibers to be innervated at one time, coming from one unit. Okay, so it just depends on the action of the muscle itself as to how many you want to occur. All right, going back, here's that acetylcholine. Here's the long term for it, acetylcholine, but ACH for short, and you're welcome to use that. I usually use ACH because it's much easier to write. So the nerve impulse, again, comes down that motor fiber. We get to the motor end plates here, and then you're going to have an interaction between the nerve and the muscle cell. And that interaction is going to have to be caused by a neurotransmitter called, in this case, acetylcholine. So they get released from those synaptic vesicles that are located in the motor neuron, and they travel through that cleft, and then they bind to little receptors on the muscle cell itself, and that causes then a series of actions on the muscle cell to allow for contraction to occur. If you look down here at this picture down here, notice we've got several sarcomeres. So the dark Z line that I'm drawing in right here to Z line, that's one sarcomere. And then to the next one, that's the next sarcomere. I have down the very center, a little review, is the M line. I have the I band from here to here. I have the H zone. Well, H zone is actually here. I don't even know if I went over that earlier, but that H zone is right here from here to here. So it's just the myosin. And then the A band goes from across all of the myosin and only some of the actin in there. And then when we have a contraction, those actin molecules or fibers move in towards each other. So from Z line to Z line, that actually gets shorter. That's a contraction. All right, so here's my nerve, motor end plate, my little synaptic cleft, my synapse. There's a lot of terms that refer kind of to the same area. Neuromuscular junction is just the junction between the neuron and the muscle cell. The synapse refers to this actual space and what's occurring in this area right in here. So there's a little bit of nuance in the difference in term, but they all really refer to the same thing. So if you call this a neuromuscular junction, I'm not going to, say, hey, that's wrong, because it's not wrong. Specifically here, we're going to talk about the action in the synapse itself. So motor end plate too is another one. All right, so acetylcholine in the vesicles have to move to the surface of that neuron. They have to be released. Oh, there's a term. Remember this? Exocytosis. That is how, see, I told you not to forget these things. They come back. That is how the neurotransmitters released from that cell. So this end yellow, this is a cell. And that when that vesicle moves to the edge, like that, it comes down to the edge, it actually binds in with that and causes a gap to open up, and those neurotransmitters then can leave. Exocytosis. We learned all that earlier for a reason. All right, so the acetylcholine jumps the gap, binds to a receptor on the muscle cell, sodium moves in, the sodium then sends a wave of actions down to the through the muscle fibers and stuff, eventually down to one of these cells. So here's my long muscle cell here. And at that point, this is where we're going to have the sarcoplasmic reticulum come into play. And I told you earlier that the sarcoplasmic reticulum holds in it calcium ions. Calcium ions were important here at the neuromuscular junction to allow those vesicles to move to the surface. And then again here, so there's multiple functions here. And then again at another place during the muscle contraction, they're going to be housed in the SR because they're going to direct some more activities. They directed the activities of the vesicles, but they're going to also direct the activities of the actual muscle contraction. All right, let's kind of get off for a second and talk about this. Myasthenia gravis, when things go wrong, right? Because they do. Myasthenia gravis is one of many different types of autoimmune disorders, but this autoimmune disorder actually targets the end result or the symptoms that you see come about with muscle contraction. You can see in this guy's eye. It affects all over the body, but it's kind of visible here in his eye. See how his eyelid is drooping? So you're having problems with muscle contraction. And what's occurring here, unfortunately, is your own antibodies that are supposed to protect you from foreign invaders, kind of like viruses or bacteria and stuff like that. They kind of lose their way. They get confused. Something occurs, and instead they begin to target your own cells or target parts of your cells. And in this case, the antibodies that you produce are actually targeting those little receptors I was talking about on that neuromuscular junction. So if you remember, here's my nerve cell, here's my muscle, and then I had these little proteins that receive the acetylcholine as it comes across. They have to have receptors that allow that acetylcholine to bind. And if the antibodies are destroying that, they're basically making that whole unit inactive. So it can't work. So if the acetylcholine can't bind, then you can't have that action. Remember, the next action was sodium moving in. If I can't have sodium moving in, I can't have the next action occur, which will cause that contraction to occur. So basically, it's a dead end. The nerve impulse comes down, the calcium moves in, the vesicles move to the surface, and boom, everything stops from that point. And if I can't get the next step to occur, there's no muscle contraction. So there's that droopiness or lowered muscle contraction because not everything's destroyed, just a lot of those receptors. And so you have less of a muscle contraction. So you can see it in his droopy eye, but it also causes droopy facial. Really, we see the face the most, but all over, you're going to have problems with muscle contraction all over your body. Somebody with myasthenia gravis really has a lot of those receptors that are missing, really about a third of what a normal person would have. So it's not 100%, that would be death, but eventually it can go to that. But it's greatly reduced. It's reduced by 2/3 of what a normal person should have. So there's going to be a lot going on with not just his eye, face drooping, muscle contraction in like the appendages and the abdomen and the heart. Think about that. The very weak and fatigued muscles because they're not getting the messages they need to contract. One of the things we call, besides that droopy eye, we have what's called a flat smile, because the muscles that control your smiling are, again, not getting the message. So there's not really this upturned mouth that you would get in a normal smile. It's just kind of across here. There's not as much movement going on in there. A nasally voice is another one. Obviously, any muscle actions are going to be problems. So you see difficulty in chewing and swallowing, can't get past that chewing, swallowing. Limbs are weak because of the muscle actions. But you got to think some of the vital things are occurring in here too. So the diaphragm and the muscles between the ribs that allow for respiration to occur, allow for breathing to occur, are also going to be affected. So there's going to be a problem with breathing, getting oxygen to your cells and getting carbon dioxide out, and it may require a ventilator at some point. There's no cure, but there are mechanisms for controlling sort of some of the symptoms and controlling the progression of the disease. There's inhibitors that you can prescribe. They can take out the thymus that has some actions on there. Intravenous antibodies that would kind of counteract the action, plasma exchange and stuff. One of the in the science community or the math community, probably not in your community, but a famous individual that had myasthenia gravis was this guy called Gregory Chinovsky. Gregory Chinovsky was a mathematician. So, I mean, it doesn't affect the brain. You got to think mathematicians very smart. He was brilliant. He and his brother took pi, pi, the number 3.14, and then it goes on forever. After that point, we all know it by 3.14, but it's really, it's a never-ending number. So mathematicians are kind of fascinated by this, this finding the end to pi. There is no end. Their job, and they work for, I can't remember, Cornell or somebody. Cornell, I think, is who they worked for. Their job was to kind of keep elucidating the next number and the next number and the next number for pi, and they took it out to over 2.2 billion digits, which was a world record at the time. Think about that number. Billion is huge. Now, these guys did this in Chinovsky's apartment in New York because he was so weak and so tired and needed to rest so much that he really couldn't even leave his apartment. So they had to take computers and over the internet buy computer parts, build a supercomputer, because they had crashed so many supercomputers that they had bought time with for these different universities. They would buy time and use their time in the middle of the night. But they crashed these computers, and they were like, nope, you can't use our computer again. So they built their own supercomputer in Chinovsky's apartment and were able to make this feat. The Japanese have come back and gone further than that, but at the time that was a record. So you can see, I mean, it causes issues, but it also, people can work around it and do things too. It doesn't really cause anything with brain functioning. It's just the muscle functioning that's the problem. Remember, because it's not the neuron. The neurons all work fine. That means the brain works fine. It's that reception of that acetylcholine across the gap that becomes a problem. So it's the muscles that are affected. All right, I'm going to stop, I think, right here for this PowerPoint presentation, and then I'll pick up with part 2 later.